Okay. Good morning. We want to welcome you to the Centers of Excellence in Iron and Heme Disorders, interviews with giants in iron. And we want to welcome you today. Today, we're going to be interviewing Dr. Harry Dailey, who is an emeritus professor in the Department of Biochemistry and Molecular Biology at the University of Georgia. Uh, Harry got his bachelor's degree and PhD at UCLA and has been like a I don't know how to put it, Harry, but a leader in heme biosynthesis, in the biochemistry of the enzymes involved in heme biosynthesis, as well as Jerry Kaplan tells me maybe a reductase person many years ago. Is that true?
I actually started that field.
See, I knew there were details that were going to come out here that would be really fun for us. And so we're going to be, with the help of Cade Leverage, who is an MD PhD student here at the University of Utah, we're going to interview Harry and learn about his foray through the field of iron metabolism. And I want to thank you so much, Harry, for participating in this interview. And I hope it's fun for us. I know it's going to be fun for me because I have lots of fond memories here. And so I'm going to stop sharing my screen. It'll just be the three of us kind of talking. Thank you again.
You're welcome.
Okay, Cade, let's do it.
Awesome, awesome. Yeah, I'm also very much looking forward to hearing about your career. Just first, we wondered if you could just describe kind of your background and your family, before you got into the iron metabolism field.
Yeah, so I have two older sisters. My dad worked on the railroad. My mom was a baker for a while. And actually, they grew up as children in the Depression. And during World War II, my mom was a Rosie the Riveter and built airplanes. And my dad built ships because he failed his physical to get into the Army. Um, so I was born in California. The rest of my family is from Kansas or Missouri. But they moved out here, like I said, during the war, and they stayed. Uh, my background is very blue collar. Like I said, my dad worked on the railroad. I'm the first person in my extended family to have graduated from college, uh, and certainly the first one to be crazy enough to get a PhD. So I still have two older sisters, and I have no other brothers. And I guess that's my background. I grew up in San Jose until I was in seventh grade, and then we moved to San Luis Obispo in California, where I went to high school, and then I went off to UCLA as an undergraduate. I was a pre-med because I thought I was going to be a surgeon, slice and dice. Um, and I became a bacteriology major because that was the easiest biology major, but then they changed the regulations and it became just like everyone else. So I actually got a bachelor's degree in bacteriology, and I'm one of the last people, I think, at the University of University of California, Los Angeles, to get a BA in bacteriology. Uh, so while I was an undergraduate, this sort of goes into how I got into the field. Um, I took a class taught by a woman by the name of June Lascelles, which is a little unusual because back then there weren't very many women in science anyway. And she's actually, I discovered later on, was a leader in the field of tetrapearl metabolism in bacteria. And I took a class from her, and I was really fascinated. And so I went to her and I asked her if I could work in her lab, which nowadays is very common to do. But back then it was a little bit unusual. As an undergraduate, I think there were only three or four in my class who actually went and worked in people's labs. So I went to her lab, and she put me working on ferrochelatase, an enzyme that I had never heard of, knew nothing about. It's responsible for putting iron in protoporphyrin to make heme. And I just really fell in love with doing lab science. And I realized so I was there in my junior year, the end of my junior year, and then that summer I was fortunate to get a fellowship that paid for me to stay in the lab in the summertime. And so I stayed for about a year and a half altogether as an undergraduate working in a lab. And that's when I discovered that I like working in the lab. And I thought about myself, and I thought about becoming a doctor, and I realized that with my personality, I would not have a great bedside manner because I would tell people what to do, and if they didn't do it, I'd probably not be the happiest camper. And also, I'm an introvert, so dealing with people is a little tiring at times. And working in the lab was something that really appealed to me because you succeed or fail on your own. I mean, obviously you have a lot of help around, you have advisors and things like that. But basically, if you fail, you've got no one to blame but yourself. And if you succeed, you can congratulate yourself some for that. And I like that. And June Lascelles, who is my mentor, was a person who really gave me a lot of free reins to do what I want to do. So uh, one day, she and another faculty member came to me while I was an undergraduate and said, "You should apply to graduate school," which I had not really thought about. And so I did. I stayed at UCLA and got my PhD 4 years later, then working in her lab, where I did work on really sort of the supply of iron for heme biosynthesis. So I looked at ferrochelatase, um, the iron reductase thing as well. I mean, there's a person by the name of Owen Jones who had shown that we knew that you need to have ferrous iron for ferrochelatase. And iron in cells is going to be, a lot is going to be ferric. The question is, how do you reduce iron? So he actually came up with an assay and showed that you could reduce ferric iron to ferrous iron for heme biosynthesis. But the assay involved using an oxygen electrode, and it is difficult. So when I was a graduate student, that was done in 1972. And so when I was a graduate student, it's about 1974 or five, something like that. I was looking at iron reduction for heme biosynthesis, and I wanted to find a way to actually measure that. And so I stumbled upon a paper that had the compound called ferrozine that everyone now knows about, that had been used by chemists as a ferrous iron trap. And so I thought, well, this might be interesting. And so I actually developed the assay using ferrozine. And that actually goes to the story with Jerry Kaplan.
I know that assay. I learned that assay maybe 1981 or 82. Could that be
Yeah.
right? Would it have been out by then?
Yeah, I was out by then. I think the paper, I don't remember the date, but it had to be like maybe 1975, 76, something like that, because I graduated in 76 and that paper came out. The funny thing with Jerry, though, is there's a thing at NIH and the retirement of David Badman.
Yep.
And he was there and I was there, and he was a speaker. And before his talk, we were just chatting. He was talking about iron reduction and using ferrozine and blah, blah. And I said, "Well, Jerry, you realize that I was the one who actually invented that assay." And he goes, "I didn't know that."
And Jerry
Yeah,
loves
I didn't know
history, so
that.
yeah.
And so he got up and started off by saying, "Oh yeah, every Dailey." So that was my contribution. That was my first contribution to thing. And we did a little bit of that when I first got my position at Georgia, but I sort of dropped that and just focused on heme metabolism itself, or heme biosynthesis, and not so much on iron. So
Uh,
I've babbled enough about that.
Yeah, that was perfect. That was perfect. So yeah, the perfect segue into, um, you'd mentioned kind of pivoted to mostly heme biosynthesis. Can you kind of give us a broad overview of, your independent labs research, maybe some of your favorite discoveries, anything you feel like talking about?
Yeah. So how I ended up in doing this for my career is sort of interesting because, like I said, as a graduate student, I worked on ferrochelatase mainly, and we purified it and also looked at iron reduction, and used to do assays using pyridine hemochromogen. So I smelled a lot of pyridine in my life because you didn't used to have fume hoods to do this. You just sat out in the open with a scanning spectrophotometer that took a few minutes, and you had to set the baseline. There are no computers then. Uh, and so when I finished my degree, I went off and I worked for a person by the name of Phil Strittmatter, working on microsomal cytochrome B5. And his claim to fame was that he was the first person to have purified a membrane-bound protein, cytochrome B5. And
Oh,
so I was very interested in working on membrane proteins. And the funny story is how I ended up in his lab. Well, first of all, I didn't want to work on heme biosynthesis. I was sick of that. I was sick of smelling purity. I was going to get into a new field. And so I came up with a list of people that I could do postdocs with and talk to June. And June was an interesting person because she dealt with everyone differently. Um, some people liked her, some people didn't. She and I had an interesting relationship. So when I went in, I gave her a list of people, and she went through the list, and she goes, "Oh yeah, yeah, huh, yeah, this is a good one. This is," and then she got to Phil Stripman, and she looks at me, and she goes, "Oh, Harry, I don't know about this one. Phil Stripman is a pretty strong biochemical investigator. Blah 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 blah. I don't know how you do." And she knew that that was a challenge to me, and so I ended up working for him, as opposed to the other ones that would have been fairly straightforward. Um, so she sort of maneuvered me into that. So anyway, I worked with him, did a postdoc at the University of Connecticut Health Center in Farmington. Um, he was also both my mentors, both June and Phil, were interesting in that they were two very apolitical individuals. And so as mentors, they were terrible in that regard because I didn't get connections. I didn't learn about making connections, but they both also worked in the lab themselves. And so I got a real feeling for the value of staying connected on your own. And also, they were very good scientists who were very hardcore. So I came to understand the value of good quality science, I guess I would say.
Hard to
And
not be at the bench, right?
yeah,
Yeah, yeah. I mean, that's what hooks you, I think, at the beginning is
yeah.
the is
It,
that
it's
fun part.
it's a sad thing, I think, because people get hooked on being a scientist by doing science, and then they end up sitting behind a desk
Writing.
and yeah, writing a lot, right?
A lot of
So,
writing.
so when it's time to look for a job, I figured I'm not going to work on cytochrome B5 because Phil's got this wrapped up and he's going to get all the money. So I looked back, I'd forgotten how much I hated the smell of purine, and I realized that very little had been done on ferrochelatase or heme biosynthesis in the previous years. And so I decided I'd go back and work on that because I figured I could get funding on that. And
So the key point there is find something that not a lot of people are going to compete. You have to compete with, but is still of interest for you.
yeah,
Yeah, yeah,
you have to have something that you have experience doing that you can do and that you have a passion to do. I mean, I'd always tell my undergraduates and graduate students, I said, you got to find what your passion is, because if
yeah.
you're not passionate about doing science, you shouldn't be in the field. Science is not an easy thing to do.
It's too
And
hard.
and if you don't love it, if you don't wake up every morning wanting to go into the lab or go to bed at night thinking about your experiments the next day, it just may not be the right field for you. I mean, it's very important to find what your passion in life is. And fortunately for me, my passion was doing science and I love coming into work. But anyway, so I came to Georgia. The funny thing was I had some interesting experiences on job application things, which I'm sure everyone does. Uh, I had one job interview that I was interrupted in the middle of my talk by a postdoc who obviously was trying to make a mark with his boss, who claimed that what I had done was not possible. And I
Okay.
said, look at the slide, there are the data. Anyway, I found out that I was at Georgia, that the committee had people who were biochemists and people who were microbiologists. And so the microbiologist knew June Lascelles, the biochemist knew Phil Stratman. So they both thought they were getting one thought they was getting someone working on photosynthesis. The other one thought that they were getting someone who worked on flavoprotein mechanisms. And they got someone who didn't work on either one of them. It worked out okay. And so I
Oh,
came here and we started off by working on heme biosynthesis and iron metabolism in what is now Rhodobacter pseudomonas, which is what my old boss worked on. June Lascelles worked on that. My first graduate student was Mark Moody, uh, who's had a very good career in industry business, and I'm very proud of him. I think he's probably about retiring now too. Um, and then we looked at heme biosynthesis. We were looking for sources of material to do purify ferrochelatase, and so we had chicken blood because there's a lot of chicken processing plants nearby, and also beef liver because I'd done a lot of beef liver stuff as a postdoc. So I knew how to get mitochondria out of beef liver. And so that's sort of how we got going. And throughout so I'm an old fart, right? I was born in 1950, and so I've been around a long time. So I've gone through starting off, we were interested in biochemistry and enzyme kinetics and purifying proteins. And then molecular biology came along. And so then it was a matter of cloning things and looking at expression of things. And we got involved with crystallography when they hired a crystallographer here on campus because we had protein. And so we got into crystallography and we also did some cell biology and we did gene knockouts and made mice and the whole thing. And it's just a trail that you sort of follow. Um, you never know when you start off where you're going to be. It's just not possible. I mean, science moves forward some by uh, our own, thoughts, but an awful lot of it has to do with what becomes available at the time. It's like I mentioned before we started the story about uh, David Sheeman, who's well known for his work and the fact that he had done an experiment where he ingested N15 labeled glycine. That was only possible because a few years earlier, N15 became available. Before that, things weren't available. And so science sort of moves forward with methodology. Molecular biology opened doors, computers opened doors. We went from looking at background noise to realizing background noise was data. And then you get large data sets. Well, you couldn't do anything. Well, now you can do things with large data sets. So really it's possible to, in my field, to sort of stay in the same general field, but to move into different areas, cell biology, animals, bacteria. I mean, I'm a little bit of an oddity because I'm a microbiologist and a biochemist. And so my lab, I have people who got PhDs in microbiology and PhDs in biochemistry. We work on bacteria. We do a lot of things on bacteria. We do a lot of things on human-related things, on porphyrias and genetic diseases. So so it's always fun for me I go to meetings and some people are biochemists or some people are microbiologists and we have different sorts of conversations. But I think it's the important thing is to keep your eyes open um, and and when new technologies come available, you don't have to learn how to do them. You just have to understand what they can do for you and make connections with someone who's an expert in that field. And that's what we did with crystallography. I mean, I can make crystals, but I don't, collect data. I don't know what to do. But those people know that. And so, I mean, that's how we learned, how to do knockouts in ES cells and make mice was by doing collaborations with other people who do those things. And so that's the thing. You have to understand that science is now a very collaborative sort of field, much more so than it was 50 years ago. 50 years ago, you worked in your lab, you were responsible for your discoveries, and actually publishing a paper with two authors on it was a real thing to try to do. Nowadays, you can't publish a paper with two authors. If you send a paper in, I don't care how good it is, you send a paper in Nature with two authors, they'll reject it right off the bat because they'll think it's not important enough. If only two people did the work, you have to have collaborations. And so you have to really be open to collaborations. And that's a good thing because you also hear a lot of different ideas from different people that make you think about your own projects.
Yeah. Okay. So, Harry, in thinking about your career, and you have mentioned a few of the individuals that have influenced you and sort of directed where you were going. In the iron community, who were the big shots that sort of influenced you?
Oh, that's I'm iron because I work on the enzyme that puts iron into heme, you know. And I think probably the person that was is an iron person was Prem Panka.
Yeah,
I met
but
Prem.
also a heme person.
Yeah, that's why I met him because he was interested in iron and heme biosynthesis. And I met Prem at a meeting and Prem was a very introverted person. I remember the first time I saw him, he's this quiet person, just sort of moving around in the background. And then he came and started talking to me and told me who he was. And I knew his papers, but I didn't know who he was. And so we actually formed um, a nice friendship. And I think the thing that to me was special for Prim, I mean, there are a lot of people. I mean, Jerry Kaplan was one, obviously, because I know Jerry. I knew him from when he was in Connecticut, and also uh, the other people at Utah. But Prim was someone sort of special because Prim had actually he was the first person to suggest that there might be two different ALA synthases, and that one of them for heme biosynthesis in erythroid cells was regulated by iron. And that was very early on, and it was widely rejected. Um, and then it became accepted. And I invited him for a seminar, and he started off someplace in the seminar, and he had a cartoon, which I always remember to this day. And there are several boxes in the cartoon. And in the boxes, there's this man, and he's standing out in this field. There's people wandering all around, and he's yelling, "It is so," and no one's paying any attention to him. And then you get to a box where, God comes down and says, looks at him and goes, "It is so," and all the people look and they see God saying "it is so," and then all the people say "it is so," but they've sort of forgotten that it was that one person that started the whole thing off. And that was Prim. And he did that actually twice in his career. He did that for iron regulation of heme biosynthesis in erythroid cells being differentiated from non-erythroid cells. And also he then did with the kiss and run
Yeah.
for iron delivery between things, which was very controversial. And there was very strong opposition to that from some people. And now it's just accepted. It's a general thing. And so I think the thing I learned from that was that you may not, what you find may not always be generally accepted, but you stick by your guns, because if you believe it and you have the data, eventually everyone will come around to it. And the other thing that you learn from that is um, you may not get credit for it. And I think that's one thing that people coming into science, I would say to them, and that is, if you come into science because you want to become famous, good luck to you. I mean, the thing for me is um, being in science and also being in academics, and I've done some stuff in administration. It's not about, my legacy. I don't think of my legacy. What I think of is I think of doing good science, asking good questions, collecting data, and sharing that. So, it's like the Ferrazine thing, long forgotten. And now, as I've sort of stopped doing research, 10 years from now, if I go to me, I'll expect that no one will remember who I am or care. I'll just be that old gray fart in the back of the room there. Um, so
Who's
I think it's really
probably the
important.
most interesting to talk to, by the way. Just telling you, Cade.
Yeah. Yeah,
Yeah.
that's always the case. Yeah.
Those are the ones you want to hunt down and just kind of pick their brains and learn. And that's part of why this interview is so fun. I just am really loving to hear the history. Okay, Kate, keep going. What else do we want to ask?
Yeah, no, I've super enjoyed this as well. So I guess coming back to your research in particular, and this might be a difficult question, but if you had to name one thing from your research or one discovery that you're most proud of, what would that be?
Okay.
One or two.
Yeah. So the thing is that um, there's a lot of different things, but I think the two things that I take the most pride in, one was actually mine, and one was my lab's. And I mean, I I think you need to differentiate sometimes. I mean, you run a lab, and the people in your lab do the work. It may be your idea, maybe it's their idea, um, but yeah, you have to share things. But one thing I actually did myself, I'll come to that last. But the thing that I think is most impactful, the discovery made was when we discovered the mutation that causes variegated porphyria in South Africa, the so-called South African variegated porphyria. And for that, that was really Tammy. Tammy was my wife who who ran my lab and allowed me to play around in administration. Uh, she may be the best scientist I've ever known, actually. She was really, as a molecular biologist, she was incredible. But we got the DNA, and it turned out there's this little girl in South Africa who had a very bad case of very good porphyria. And we got the DNA from Pete Meissner, who's a longtime collaborator and friend. And so Tammy actually found the mutation. This was back when sequencing was not easy. Now you just send things out that get sequenced. Back then you had these big gels that you had to run things. And so actually finding mutations was a lot of work. And so Tammy found the mutation. She actually found two mutations. And turns out one was from the little girl's father, who was German, and one was from the mother, who was Afrikaan. And that was the South African very good porphyria mutation that goes. It's a fascinating story about that. It goes back in the 1600s. And so we went to South Africa, and Tammy actually did the presentation on that. We met the little girl and her family. That was really a lot of fun. And for us, it was interesting, and for South Africa, it was a big deal because this is the second most commonly inherited genetic disease in South Africa. It affects over, at one time they said, I think 40,000 people or something. So finding the genetic basis for it was very important because it turns out that it introduces a restriction site. So they now can screen newborns by just taking cord blood and doing a restriction digest and see if they got the mutation. So if you have the mutation, you know it early on, you can deal with the disease. So that was really nice. And it was a big deal for South Africans because of that. So Tammy actually was the one that did that, but she said, well, this is really important to Pete. And so Pete's actually the first author on that paper because Tammy said it's more important to him than to her. And she's a second author. And for them, it was. I mean, they got TV interviews. They were in the Sunday newspaper. Big time stories. So that was probably one of the most significant discoveries we made. The
And you
other one
can
was
feel
actually,
it still, right? I mean, you still feel sort of that discovery and the excitement of it from that long ago.
yeah, well, it's fun because I mean that's one thing we did that actually had an impact on human populations, fairly significant impact. It doesn't cure the disease, but now we know what it is, and
And you
so
don't know
that's
what it'll lead to.
yeah.
Yeah.
So the other one is actually was my work, and I had a lot of fun with that because um, I mean, one thing I always like doing is trying to find new enzymes, and so the heme biosynthetic pathway, everyone knew what it was. Okay, we all knew the enzymes, we all knew the steps. Other than the first step, everything was the same. So then we start getting genomes from different organisms, and we start finding that there are sort of things that are missing. And so one of the things that we did in my lab was we looked for the missing enzymes, and um, so we actually identified. I mean, we protoporphyrin oxidase. We found the first enzyme, which was named heme Y. But then when genomes came along, it became clear that heme Y was not present in very many things. Um, and so, like gram negatives didn't have any heme Y. And so we then discovered that there were two other enzymes that did it as well. And as a graduate student, also Tammy worked on that in the lab. So we designed, we discovered, the heme G in E. coli was a different enzyme than the heme Y in Bacillus that we found. And then there's another enzyme that we called heme, what was it? I forgot it was heme J. Sorry about that. I forgot the initial.
I love that you have the chart to go look at.
Yeah, I do because one thing we wrote a review on heme biosynthesis after we finished this thing I'm going to tell you about. And so in order to make sense of it, we had to rename all the enzymes of the pathway. So anyway, so going back to, so we found that there were three enzymes that did protoporphyrin oxidase activity. And so then we looked to see on coprogen oxidase, which there's an aerobic form and an anaerobic form. And the aerobic form was known, but then when the genomes came out, it turned out that very few organisms actually had that. It's called heme F, had heme F. And so we were looking in, but everything seemed to have heme in. but it turned out that everything annotated as hemin wasn't actually a coprooxidase. They'd just been misannotated. Actually, 3/4 of the hemin annotations are not for hemin; they're for other things. And we sort of discovered that. So we started looking for, and we couldn't find the coprooxidase in gram-positive bacteria. And so we looked and we looked, and so this was something that fortuitous, a person by the name of Svetlana Gerdes contacted me about doing some annotations. That's what she did, and we ended up talking, and she and Tammy started looking through genomes and trying to find the missing coprooxidase. So it's a long story, but she they then came up with an enzyme we call heme-Q, which was essential for heme biosynthesis in gram-positives, but we didn't know what it did. It wasn't coprooxidase. And so that sort of sat there, and that bothered me. And so I actually started working on this myself because I didn't want to assign something to a student that I didn't have a clear vision that there's going to be an outcome to it. And so I worked on this, and it's sort of fun because I go out in the lab and I work, and the students come out and say, "Oh, what are you doing?" I'm going, "I'm just running some assays, just running some." So I didn't tell anyone what I was doing because I thought if I told them, they thought I was crazy. Because my idea was that the pathway wasn't conserved in gram-positives, that something was different about it. And the tip off on that was I went back in history. So I went back to a paper in 1956, in which they said that in this gram-positive organism, they didn't ever see protoporphyrin, but they saw coproporphyrin, a lot of coproporphyrin, and also saw some coproheme, but obviously that was an artifact. And then Nick and Judy Jacobs did a lot of work back in the 70s on gram-positive and gram-negatives, and they always found that gram-negatives could make lots of protoporphyrin and heme, but on gram-positives, they never found any never found any in protoporphyrin, but they always found coproporphyrin. And so I had this idea, I'd go, what if the pathway is different? And so I started working on it, and the end of the story was it is different. So in gram-positives, you actually go through a slightly different pathway. You make coproporphyrinogen and you oxidize that to coproporphyrin. You put the iron into coproporphyrin instead of protoporphyrin. So you now have coproheme, and you convert coproheme into protoheme, and that's the enzyme heme-Q that we had discovered. So that was a new enzyme that had been annotated as a totally different enzyme, and so we discovered that there was a different path. And so I did that with my own two hands, and it was a lot of fun. And so I think that's for me that was the most fun because I can look back and I go, it didn't make sense when I got started, but then after I did it all, and I went back and I read all the old papers, it makes a lot of sense. And what had happened was we had been trying to fit, a square peg in a round hole because there were square holes that we all knew about, but we were trying to fit the data we got. We knew that heme Y in gram-positive organisms actually oxidized coproporphyrinogen better than it oxidized protoporphyrinogen. But we said, oh, that's just an artifact. And we actually never looked at ferrochelatase in gram-positives because we knew that ferrochelatase didn't put iron into coproporphyrin. But had we looked in gram-positives, which I then did, we discovered that. And so that was a lot of fun because that project also ended up involving, John and you guys out there at Utah.
Yeah, John Phillips.
Because you guys were set up to do the porphyrins, and I didn't want to trust myself just, saying, oh, yeah, this is what it was. So we'd do our assays and send stuff out to John. So that was
another important collaboration.
Yeah. So that was another fun story. So, and yeah, those are two things that I look back on. I mean, we did some other things, and I'm very proud of things some of my students did, but those are things that come to my mind.
Great. Go for it, Kate. There are just a couple more questions we wanted to cover.
Yeah. Both very cool discoveries. I guess I kind of want to combine two more questions, which is first, in your mind, what is, or I guess for you personally, what is the biggest unanswered question you have in iron
And
or
it can't
heme.
be the God question, by the way.
And I think in that vein, as you know, few scientists are moving into iron and heme research, what advice would you have for them?
Okay, so going to the first thing, I actually thought about this a little bit, after I talked to you initially. And I actually think that the biggest question in both heme and iron is heme and iron metabolism in the brain. I mean, I go back to so when I first got into science and went to the first Gordon conference, I remember arguments between people saying there's only one ALA synthase. That now, and in their papers published saying there's only one positive data, and turns out there are two. And heme biosynthesis regulation is different in erythroid cells than it is in other cells. But in my early days, it wasn't. It was all, it was a uniform sort of field, and you sort of squished everything into that. And then people started looking at different organs, and they go, well, you got erythroid cells, and we now know that's much different than in hepatic cells. And honestly, as far as we know, we assume that every cell makes its own heme, but we now know that heme gets moved around, and red cells during early development actually overproduce and excrete heme. Where does that go? So heme probably gets moved around. But I think that I mentioned the brain because we know very little about. I mean, it's not a red organ, right? So if you're looking at iron and heme, you look at red things, and brain is not red, but yet we all know that carbon dioxide is a signal molecule, and the only source of carbon dioxide is breakdown of heme. So if carbon dioxide is a signal molecule in your brain, then maybe we're not making, maybe the brain makes heme, but it doesn't make heme as heme. It makes heme as a precursor molecule. I mean, I think you always think of in heme biosynthesis, you think, yeah, heme is the end product, but and everything else is a breakdown product, as opposed to thinking of heme as an intermediate in a pathway.
Which
Um,
is becoming more actually prominent in the
yeah,
field. For me, at least as I've been reading things, the breakdown of heme. I like the idea of it being an intermediate. I really like that, Harry.
yeah.
Yeah.
Yeah, in photosynthetic bacteria, you make all these, phycobility proteins, and those are all heme breakdown molecules. So,
So we
uh,
should have known.
yeah. I mean, so I think what happens, and I think advice that I would give people, just in general, is don't get stuck on dogma. Question everything. I mean, I had graduate students out, they come to my lab and they go, oh, I thought of this and thought of that. And I go, I don't think this is going to work. And I go, try it. You have your project, keep doing it on your project, but you can try that. And if it works out, that's going to be great. And if it doesn't, okay, you've tried it. And there have been times when students have come up with ideas that worked out. Um, and so and they were sort of off the wall kinds of things. So I would say, yeah, there's a lot left, and don't settle for the current dogma, because I mean in my lifetime I went from having a pathway to heme. I mean, early on there was one way to make ALA synthase, and then later on, oh, there's actually two ways, and plants and most bacteria make it in the other way. They don't make it the way we make it. And so that then opened up the field. And then there are two ways to regulate ALA synthase and even mammalian things. And we have no idea. We really have no clue how heme biosynthesis is regulated in most bacteria. And even within our own bodies, we know about liver, we know maybe a little about kidney, we know some about erythroid cells. I don't know anyone that works on brain. I don't know if NIH will even fund brain stuff. I mean,
It's a
um,
good question.
so it's that. And the other thing I'd say is, to new people, find your passion. Um, if it's heme, then stick with it. Realize that you may not win a Nobel Prize. Uh, you may have a hard time getting funded, but you gotta, believe in yourself and what you're doing and work hard. Um,
And love what you're doing. That makes the work easy in the sense of you want to go in even when it's hard.
yeah, it's science is not easy.
No.
Uh, but it's rewarding. I
Yes, it
think
is.
that's the
Yes,
most
it is.
important thing.
Well, I for one am hopeful that in a meeting in 10 years I'll see you and I can go and chat with you and keep learning from you. I've really enjoyed this conversation.
Well, I mean, the thing I'd say is, I mean, I would love to meet you at a meeting. When I was a newbie, when I first went to Gordon Conference, I think I was one of maybe two graduate students there. It was much different. It was the big name people. I mean, David Sheeman was there, you know. I didn't walk up to David Sheeman. I'm not going to walk up to David Sheeman and start
Yeah.
talking to him because he's so important. And the funny thing, my first real conversation I had with him was I was at another meeting and he came up to my poster and started
Mm.
talking. And I was so sorry I hadn't talked to him sooner. These fascinating conversations. And that's happened with a few different people. So um, I I go to meetings now and you know you age on the outside, but internally, you know, I don't think of myself as being old like I am. And so I go to meetings and I have students come up to me and go, "Oh, Dr. Dailey, can you talk to you? Have time to talk to me?" And I go, "Of course I have time to talk to you." And I'd say to Tammy, I go, "What's with these people? Why don't?" And she says, "Well, they're impressed by you." And I go, "Why? I don't, why are they
You're
impressed
Harry
by me?"
Dailey.
I mean, I mean, this last meeting I went to, this person came up and said, "I said thinking about asking you for an autograph," and I'm going, "Why?" You know, so you know, I'd say yeah, I love talking to younger people because they have you talk to old people and they get sort of stuck in things. You know, they've read papers, they know what's going on. Whereas young people have crazy ideas. And so it's crazy ideas that drive science. You know, it's not old ideas. And so it's always fun to talk to people and to ask them questions and so yeah, I just encourage all graduate students and postdocs and junior faculty to never be afraid to go up and talk to someone. And if they turn out to be a jerk, that's their problem, not yours. You learned a lesson, but I'd say that, you know, my experience is 90% of the people you're going to walk up to are going to be happy to talk to and spend time talking and maybe go have a drink with you. Um, so yeah, that's my advice.
Well, that's good advice. I'll try to put that into practice.
Well,
Do so.
Harry, we want to thank you so much. This has been fun. It's going to make my day and maybe my week so that I can smile as I'm thinking about the important discoveries and the good people in the fields of iron and heme and the collegiality that exists and
Yeah.
we want to continue to have persist through this community. So thank you again for this interview. It was great. And we'll uh, probably go in and we're recording this. We'll save it and then we'll get it posted up on our YouTube channel, but also on the Center for Iron and Heme Disorders so that people can go. We'll have a little section that will be learn some history in iron and heme kind of
Yeah,
thing.
Jerry will look at it just to see if I mention his name.
That could be true. And now, we'll give him time when he gets interviewed so that he can respond to your
Yeah,
comments.
I don't think Jim Cushing would care if I mentioned his name or not, but I probably should have mentioned Jim's name long ago. But
Jim is living in New York
yeah,
and is in his I think late 80s.
no,
Um,
I think Jim's I think maybe well maybe uh, playing the piano like Beethoven, deaf playing the piano.
in, and that's 100% true. He's a really concert pianist level
Yeah,
skill set.
yeah. We did mention, but he was he was an incredibly uh, impressive person to me, and fortunately we started talking early on. But Jim's mind is like I remember going to meetings and Jim would ask a question. He said, "Well, I remember back in this paper back in 1957,
Yeah.
you know, and named the authors of the paper." And I'm going, and when I first heard him, I'm going, this guy's just bullshitting us. I mean, he doesn't know, but he knows this stuff.
No,
He
he
knows
does.
this stuff. And
He
it's
totally
so
does.
fun to sit down and talk with him. And at Gordon conferences we got where he and John Phillips and I would share some gin and tonics and sit around and say things about people and stuff. And so
Just
I miss
make
it.
community.
Yeah.
That's
Anyway,
awesome. Again, thank you so much, Harry. And Kate, thanks for doing the interview. And we'll keep you posted, Harry, on when things get posted. We'll probably post them all as a group so that people can kind of surf through them. But
yeah.
this has been terrific. And
I
tell
want
Amy
to listen
hello.
to the other ones too.
Yeah, tell Amy hello for me.
I
Um,
will.
and sorry I missed the Tetrapyro conference this year.
Yeah.
Yeah, it's a great Gordon conference, by the way, Cade. It's a just, and the mix of people, really great biochemists and also bacteriologists, microbiologists. It's a good Gordon conference. I highly recommend
It's
it.
a fun group.
Yes,
Okay.
it
Well, I had
is.
fun doing this.
Yeah. So thank you so much again and take care, everybody, and we'll talk to you soon.
Okie doke. Bye-bye.
Okay. Bye bye.
Nice meeting you, Kate. See you at the meeting.
Good to meet you.